The human body is composed of roughly 37 trillion cells, and within each individual cell, genetic information is stored in the form of DNA. DNA is a long, double-stranded molecule that contains genes which encode instructions for building proteins that are essential for cellular functions. Remarkably, every cell type in our body contains the same DNA, meaning neurons, muscle cells, and even skin cells carry the entire set of genes required for the organism's development and survival. The key to their distinct structure and function lies in the fact that each cell type follows a unique pattern of gene expression. This means that genes responsible for muscle contraction are predominantly expressed in muscle cells, while genes involved in neurotransmitter production are expressed in neurons. So how does this happen? Well, the first step in gene expression is transcription. Transcription is the process in which a gene's DNA sequence is transcribed or copied by an enzyme called RNA polymerase to produce an RNA molecule. There are three major classes of RNA: messenger RNA, transfer RNA, and ribosomal RNA, of which messenger RNA or mRNA acts as an intermediary, carrying the genetic code from the DNA, which is a site of information storage, to the ribosomes, which are the sites of protein synthesis. The nucleotide sequence of transfer RNA and ribosomal RNA does not encode proteins, but they still play a vital role in protein synthesis. The sequence of DNA involved in transcription of a single RNA is called the transcription unit. It consists of the promoter, the gene, and the terminator. When there is a need for a specific gene product in a cell, transcription is initiated by the binding of RNA polymerase to the promoter. A promoter is situated upstream of the gene and carries information determining the starting site for transcription and also determines which of the two DNA strands is transcribed. It is important that only one of the two DNA strands is transcribed, because if both strands were simultaneously transcribed to produce two RNA molecules, these molecules would be complementary, leading to the formation of double-stranded RNA, which cannot be used for protein synthesis. The DNA strand, which is used as a template for mRNA, is called the template strand, and the other strand is called the coding strand. Even though this strand does not code for proteins, it is named so because it contains the same nucleotide code as the mRNA strand, except that uracil and thymine are interchanged. Note that RNA polymerases cannot recognize promoters on their own. They need the assistance of additional proteins. In eukaryotes, these proteins are many and collectively called transcription factors. In prokaryotes, this assistance comes from proteins called sigma factors. Bacterial cells have various sigma factors, each recognizing the promoter sequence of different genes. When a sigma factor binds to RNA polymerase, it facilitates the efficient recognition and binding of the enzyme to the promoter sequences, thus initiating transcription. To access the template strand, RNA polymerase continuously unwinds the DNA double helix ahead of the polymerization site and also rewinds the complementary DNA strands behind the polymerization site as it moves along the double helix. This locally unwound segment of DNA is called the transcription bubble. With the DNA template exposed, RNA polymerase starts synthesizing RNA by incorporating complementary ribonucleotides to form an RNA chain. Once the enzyme has synthesized the initial 8 or 9 bonds, the sigma factor is released and the process enters the elongation phase of RNA synthesis. Much like DNA polymerase, RNA polymerase constructs a chain of nucleotides in the 5' to 3' direction utilizing a DNA template. However, instead of using deoxyribonucleotides, RNA polymerase joins ribonucleotides to form the chain. The difference between the two is that ribonucleotides contain a hydroxyl group attached to the 2' carbon of the ribose sugar, whereas deoxyribonucleotides have a hydrogen atom at the same position. An additional distinction is that when encountering adenine in the template strand, RNA polymerase incorporates uracil into the chain instead of thymine. Thymine and uracil are structurally similar. However, thymine contains a methyl group at the 5' position. These variations stem from the fact that RNA molecules are designed to be more chemically reactive, unlike DNA molecules. DNA's primary function is to preserve genetic information, and this requires chemical stability, while RNA has a more dynamic role and requires greater chemical reactivity. This is also the reason why RNA molecules are single-stranded, unlike the double-stranded DNA molecules. Single-stranded RNA can form diverse three-dimensional structures which facilitate its interaction with different proteins needed for protein synthesis. During elongation, approximately 40 ribonucleotides are added to the nascent RNA chain every second. The RNA chain is displaced from the DNA template strand as RNA polymerase moves along the DNA molecule, and this process continues until RNA polymerase reaches the end of the gene where it encounters the termination sequence. The termination sequence consists of sequences that incorporates a GC-rich region and multiple uracil nucleotides into the RNA chain. Since guanine is complementary to cytosine, the mRNA form a structure called a hairpin loop which temporarily pauses the RNA polymerase. This pausing, coupled with the weak adenine-uracil bonds, cause the mRNA to fall off. This mode of transcription termination is known as intrinsic termination. In some cases of bacteria, transcription terminates due to the interaction of a protein called Rho with the nascent RNA chain. This mode of transcriptional termination is called the Rho-dependent termination, in which the Rho protein binds to a specific region on the mRNA called the Rho utilization site and moves along the RNA in the 3' direction till it reaches the site of DNA-RNA hybrid duplex and separates the two molecules. In prokaryotes, these RNA chains are fully functional and ready to be translated into proteins, whereas in eukaryotic cells, these RNA molecules have to be exported from the nucleus to the cytoplasm. Hence, eukaryotic RNAs undergo additional processes called post-transcriptional modification, which I'll be discussing in the next video, so stay tuned.